Hello, everyone, and welcome to Dana-Farber's Department of Data Science Frontiers in Biostatistics webinar. My name is Erica Fike, and I'm the Program Manager for the department. Before I turn it over to our organizers and speaker for today, I'd like to point out a few things. The webinar today will be recorded and will be available on our YouTube channel within the next week. You can find our channel at www.youtube.com/DFCI Data Science. If you have a question for the speaker or the organizers, please use the Q&A tab. Our faculty organizers will manage the Q&A session during and after the talk. And with that, I will turn it over to Hajime Uno.
Okay, so welcome to the Frontiers in Biostatistics seminar. So I'm Hajime Uno, one of the organizers of this seminar series. Today we have Dr. Keaven Anderson. Dr. Anderson is a scientific associate vice president, methodology research at Merck Research Laboratories. So he received his PhD in statistics from Stanford University. He did postdoc fellow for a couple of years at the Harvard School of Public Health. Then he participated in several cross-farmer working groups. The NPH, non-proportional hazard working group, is one of them. Statisticians from over 15 companies got together to address the issue of the non-proportional hazards for design, analysis, and interpretation. And he has been one of the leaders of this group. And in addition to non-proportional hazard stuff, he has been working on various methodological research projects, which include group sequential design, sample size re-estimation, and optimal two-stage adaptive design, and so on. And he's also doing an associate editor of biometrics. And then today, he will give us a talk about group sequential design with time to event outcomes, especially when delayed difference pattern is expected. So which is often the case of immune therapy trials. So when we design an immune therapy trial, so we are not sure if we can use the conventional approach, assuming the proportional hazard. So today, Dr. Anderson will share his thought and also give us some solutions to this problem. So okay, so now please welcome Dr. Anderson. Dr. Anderson, please take it away.
Thanks for the introduction. I will say just a little bit. I arrived for my postdoc at the School of Public Health and actually arrived at the Farber in the fall of 1982. And I shared an office at that point. I think I and Diane Finkelstein and Louise Ryan all showed up on the same day to share an office for a while, but we also had offices over at the School of Public Health. And after a couple of years, I decided I was psychologically unfit to be in academics, and I went off to the Framingham Heart Study and then later to the pharmaceutical industry. So none of this should be considered to be a position of Merck and Company, my employer, and all the errors are I will take full responsibility for. I usually put an abstract at the beginning just so I can remind myself what I promised to say, but I don't need to repeat it here. So my primary focus today is just going to be testing with Logrank. I just had a discussion with Professor Uno, and it's kind of what we're stuck with the FDA is pretty much use Logrank in the Cox model. So, what are you going to do if you're planning trials with those as primary analyses? And you're anticipating potential delayed treatment effect. So I'll start off with several published examples, mostly with Keytruda, to kind of show you the issues that are recurring for us and why this is of interest. And what I've been focusing on the last couple of years, and I've gotten some tools together for, is using kind of an average hazard ratio, and and then applying it for asymptotic trial design. So we'll get into those details. And it's a pretty simple extension at the end of the day to standard group sequential theory. I like to think of it just as instead of a Brownian motion with constant drift where you're choosing some points along the Brownian motion. Now basically you've got a drift that's changing over time. probably in a smooth fashion. So I'll show you that. And that really does very little change to group sequential calculations in terms of boundaries. In terms of the programming, it's not big changes. In terms of how it affects bounds is another thing. So I'll get a chance to say a little bit about that. But practical considerations that come up, over the years we've been doing this, we've kind of changed our feelings about timing of analyses, about overspending at interim analyses. So I'll talk about a couple of example designs to try and illustrate some of the issues there. And I will try to leave some time at the end, in case there's some discussion. I know there's a fairly large group here. So just very briefly, alternate things, other things I'm interested in are alternate statistical testing, so weighted log ranks, and I could give you opinions on that. I'll give you one predicted mean survival result today. We've done a lot of work with combination tests, combinations basically of weighted log rank tests in the non-proportional hazards working group. I won't talk about that today. I'll say a little bit about the graphical approach to multiple hypothesis testing, because we've tried to hedge our bets by allocating type 1 error to multiple hypotheses. And that creates some interesting challenges for group sequential design, where you're looking, say, at spending for multiple hypotheses and you want to do your analysis for multiple hypotheses at the same time. Another area related to that, which Cyrus asked me to present on later this year, is accounting for correlations between different tests, whether they're related to nested populations or intersecting populations, or as he's done with multiple arms versus a common control. And Cyrus and I both share a passion for software development. Mine is open source in R, and he's made actually a wonderful business out of it with Scitel. So anyhow, a little bit of background. I won't talk too much about this trial. Keynote 40 was a recurrent head and neck cancer trial. And you can see at the end of the day, for about 4 months, the survival curves were very much overlapping between a control. This is a chemotherapy control versus an immunotherapy alternative. So it's a pretty daring thing not to do the immunotherapy as an add-on, but somehow it worked out, but we had this delayed effect. But at the end of the day, this peak will 0.0161. It was statistically significant, but kind of borderline. So that's kind of the danger we're in of having results like this, and they recur over and over again. I have 2 trials with small cell lung cancer. One is the Roche and Genentech trial, and I'm just showing it. It has a little bit better result than the Merck trial, but basically you see this early period for about 7 months where control is actually favored over experimental. No, it pretty much is just a delayed effect again. With the Merck drug, they actually cross. But you can see there's a delayed effect. And I'll just stop at that. This looks very similar qualitatively. The hazard ratio at the end of the day was 0.8 instead of 0.7, which was seen in the Roche trial. And this one was very borderline non-significant. So, not really completely accounting for things was problematic. We went to the FDA to say, well, we really don't want to do our spending for interim analysis based on information fraction. We want to limit our interim spending so that we have sufficient type 1 error for the final analysis to have a less stringent bound. And the FDA said no to that. They were resistant to what we proposed, which I'll talk more about later. And that ended up being kind of the difference between a just missed and statistically significant trial. So how you do spending and how you set up timing of analyses is something I'll talk about later. I also wanted to just show RMST in this case, and I can't remember what the cutoff was for this, but basically there's 1.2 month difference. So, you know, not wonderful in terms of a big treatment effect, 0.8 or 1.21 months, but nonetheless, something that's potentially, you know, meaningful, especially if you think the tails of the distribution are different for better long-term results. Delayed effects occur not just in oncology. I spent a little bit of time looking at old cholesterol lowering studies. So you can see from the mid 90s, kind of first generation statin therapies for under age 65, there may be a couple years delay before the survival curves start to separate, or if you're in my age group over 65 years, maybe you know, you start to see a benefit after a year or so. Anyhow, delayed benefit is not just an oncology phenomenon. On the other hand, there are cases where we can see something that looks like proportional hazards. This is kind of a motivation for some of what we did with combination tests, where we don't want to lose power under proportional hazards. This was an add-on to chemotherapy, so it's chemotherapy versus chemotherapy plus. And you can see something that looks like proportional hazards. Maybe there's a little bit of, you know, overlapping at the beginning, and maybe that first 4 months is just lucky, or maybe it really is pretty close to proportional hazards. But in any case, this actually turns out to be the trial that you know, drive several $1 billion in sales for Keytruda or pembrolizumab. So we're very happy with this particular result. And this was actually at the first interim analysis. So, you know, you didn't have any problem with the delayed effect kind of requiring you to do a long duration of follow-up. Okay, now here's a more problematic one. And this is one that was not statistically significant for advanced gastric or gastroesophageal junction cancers. So, you know, to me, the quote we hear from the FDA is, it doesn't matter if you're statistically significant, if it's not a clinically significant difference. I think Merck has never felt like this one quite reached the something we would claim would be clinically significant. On the other hand, if you go back and do a post hoc analysis with some of the log ranks we're looking at, you can see you can get a pretty small p-value. So you'd say, well, is that a problem with the test, proposed test? This weighting scheme for a Fleming-Harrington test probably downweights more than I would find desirable. But this is one of the complaints about weighted log rank tests, that maybe some of these things show up that are significant that shouldn't be. Now, the FDA would like to have perfect correspondence between statistical significance and clinical significance, but you know, that makes their job easier. They can just send the statistician up from the FDA to say, nope, it didn't cross the bound. We like it to be a little bit more difficult for them to establish statistical significance, and then you can have the discussion about whether something is clinically significant. And you know, there, even for this, there may be some patients that like the tail of the immunotherapy curve, and they may like the potential for avoiding side effects of chemotherapy, so they're willing to take the risk. And then it becomes kind of a personal utility function as to, you know, whether or not this is a clinically meaningful difference. In any case, not something that Merck cared to take forward and propose with the FDA. This one I'm just going to skip. It's kind of a separate issue I'm not going to talk about. Now, this is the most absurd example we have at Merck of testing multiple hypotheses in a single trial while controlling type 1 error for all the different tests and also doing interim analysis. So there are two endpoints. There's progression-free survival and overall survival. You can see that across the top here. There are two experimental groups. There's monotherapy and there's combination of chemotherapy plus monotherapy, or chemotherapy plus immunotherapy, both versus a common standard of care. And then the team wanted to hedge their bets by saying, well, maybe it's only going to work in the very biomarker positive group, or maybe it'll work in the broader, even weak positive biomarker group, or particularly in the chemotherapy add-on group, maybe it'll work for all subjects. So they dreamed up this graphical multiplicity scheme. I don't think we'll ever do one this complicated again. We had a clinical vice president who was particularly adventurous in hedging his bets. But you can imagine if you've worked on these trials that doing an interim analysis for this and checking all the possible ways to reach statistical significance would be a bit of a nightmare if you had to go through it one at a time. It turns out you can use, if you're familiar with repeated p-values, there's a simple extension of that called sequential p-values. But you can just plug those sequential p-values into a graphical multiplicity algorithm, and it'll spit back at you which hypotheses are rejected and which are not. And then it's very easy to go through and check boundaries for the individual ones just to confirm that. So it's nice to have some automation for some of these things. And then more recently, you can see that there's a lot of overlap, built-in correlations between different populations and different controls. So there's a lot of potential for potentially relaxing your bounds a little bit by understanding all the correlations between endpoints. So I'm not going to say more about that, but I just wanted to bring it up as an area of interest. But it also creates issues that I am going to talk about is, well, how do I set up a time to do analyses when I'm looking at all these different hypotheses? And then if I do that, how am I going to set up spending so that I don't run out of alpha for some hypotheses before others? So those are some of the issues that come up when you start doing multiple testing. It's more common that we would have 3 to 6 hypotheses rather than 14 as we have here. Anyhow, so summary of issues. I'll try to keep track of my time. What's the impact of potential impact of delayed effect? I'll just focus on Logrank today. Sample size can vary considerably based on duration of follow-up because your treatment effect kind of depends on having a good follow-up. How do you set up timing of analyses, futility bounds? I won't talk about updating to bounds, and I won't talk about multiplicity adjustments. Okay, so I'm basically going to use a piecewise model. So I'll assume a piecewise constant enrollment, piecewise exponential failures, piecewise exponential dropouts. And then I'll look at computing an average hazard ratio and as an asymptotic approximation. And I'll show you one simulation that's representative pretty much of all others I've done to show that the asymptotic approximation really works pretty well. So you don't need to run a million simulations to do these designs. So there's no requirement for proportional hazards. We've done weighted Logrank and combination tests in the software we've built, but I'll just stick with Logrank today. So this is just an example of piecewise constant enrollment. So here I'm just saying 5 per month for 6 months and then 20. So you can see, there's kind of a change in slope. And the expected enrollment would be 12 months. So, the actual enrollment duration is random for the way I've set this up. And then for a simple non-proportional hazards example, I'll use this for the rest of the presentation. I'll just say, I'm going to design assuming there's no treatment effect for the first 4 months. And then after that, there's a hazard ratio of 0.6. And my control group is just exponential with a median of 15 months. So that might be representative of survival distribution reasonably well for some chemotherapy controls. Okay, but the piecewise exponential is quite flexible. Sometimes your control, when I've done smoothed hazard rates on survival for chemotherapy, sometimes they look like this. There seems to be, reduced failure rates early in follow-up. And then there's just this horrible, onslaught of progressive disease within a few months. And you see a very high, death rate or failure rate for progression, between 6 months and a year. And then after that, probably you're getting to the point where you have, a lower risk population continuing. And the piecewise, you can see I have very few steps here. So maybe you think that doesn't approximate so well. But when you look at the survival curves, they're pretty well overlapping. You might say, well, I could use one more point between 1 year and 2 years to get it approximated. So the piecewise exponential doesn't restrict you to any simple shapes. Okay, so average hazard ratio. Basically, it's kind of a geometric mean hazard ratio, and Perloy Mukherjee and others published a paper recently, but they cite a longer literature where basically you're looking at the log hazard ratio and weighting by the expected events. So here, if I've got a hazard ratio of 1 from 0 to 4 months, and my expected events are D1, then I get, D1 times log of 1, so I get 0 for that, and I get D2, the expected number of events after 4 months from enrollment, times log 0.6, and so it's just a weighted average weighted by the number of events and exponentiated. So basically this average hazard ratio, again, is kind of a geometric mean. So hopefully that gives you some intuition as to what this average hazard ratio is. And, for me, that makes it a little bit more interpretable. You may disagree, which is fine. So for the model I had, I said, okay, I'm going to enroll at a constant rate over 12 months and have a very small dropout rate. Control exponential with median 15 months, and then this hazard ratio of 1 followed by 0.6. And so the month on the graph here is the month after start of study. So for the first 12 months on average, you're enrolling patients. So you can see there's a little bit of a bump maybe starting at 12 months. But basically you see for the first 4 months of the trial, your expected average hazard ratio is just 1 because nobody's gone beyond that 4-month period where the hazard ratio is 1. And then it kind of has a steep drop until it starts to stabilize, maybe around 24 months, but it'll continue to go on after that. So in any case, it looks like probably you'll want your full trial to go on for, say, 24 to 36 months. And I'll plan it for 36 months.
Dr. Anderson, sorry for interrupting you. So we have a couple of questions here.
Sure.
So do you see the Q&A box here, or I can read it?
Why don't you read it?
Okay, so one question from LJ is for piecewise exponential, how do we choose the cutoff values?
Oh, well, this is just generally what I do is, I'll go back here. Why did I choose 4 months? Well, and this is post hoc, so this is after the fact. When I look across all of our monotherapy trials, and I don't know if I've gone back and looked at ipilimumab, so that actually would be interesting. But typically, what you see is a time period around 4 months. And then for the control, I've pretty much tried to match historical data, tried to make the curve look like it. So for instance, for neoadjuvant breast cancer, where you potentially have patients who are cured or at least approximately cured, they have very good long-term survival. You can use a piecewise exponential to approximate that and then make some assumption. But for the piecewise, you have to use judgment to some extent. But if you're assuming proportional hazards, you're just assuming it's 0. And so no matter what you do, you're making a choice. And so it's a little bit arbitrary, and it's a question of what do you want to protect yourself against? But it's a great question, and one that everybody should ask at the beginning of a trial. And there's no perfect answer. If you knew the answer, you wouldn't have to do the trial. Anyhow, next question.
So next one, I think it's pretty much similar, but and how do we know the choice of 4 months would do the job at the end?
Yeah, well, you don't. And it's kind of a question of what do you want to protect yourself against? And, to some extent, you want some, let's say that you think, 0.7 or 0.8 for your hazard ratio at the end of the trial is going to be what's required to have a commercially interesting outcome. Then to some extent, it's a cross between what do you believe and what are you willing to invest in terms of how good the treatment effect should be. So it's a great question. We don't want to invest huge amounts of money in mediocre treatments because they're unlikely to provide enough benefit to create much interest. So that's part of what's going on here. I mean, that is definitely an art, and we could talk about it for a long time. So
Okay, thank you very much.
That's adequate. Okay, so here I've kind of justified how long I think I want my trial to be. And so if I want to look for futility, well, sometimes we've seen a hazard ratio greater than 1. You saw that for that one trial. And we did have one trial that stopped very early with a very large early hazard ratio. And so I'll talk a little bit about, well, maybe you'll want to do a futility analysis somewhere in here. I don't know if you can you see my cursor? Or not?
Yeah, we can see, yeah.
Okay. But then if you want to do efficacy, maybe you'll want to wait till out here. I think for this, my examples, I'll propose them at 20 months, 28 months, and 36 months, just to kind of get adequate follow-up to have a reasonable chance of a positive trial if the effect is delayed. But also, I'll want to ensure that I get my follow-up so that I've got data out, late in the curve, so that if there's a substantial portion of the benefit, whether you're looking at the Kaplan-Meiers or RMST or average hazard ratio. If the real benefit is out in the tail, you want to make sure you get enough data that becomes apparent. Okay, so here in this particular instance, if you have 100 patients enrolled, you need about 35 to 40 months until you get 65 to 70% who have events, which seems to be what both the EMA, European and US regulators would be looking for in a advanced cancer indication. Now that may not be, that wouldn't be the case for something again where you'd have a high cure rate. But this is often the case for metastatic disease in our experience. Okay, so now this is just saying, okay, if I did that, chose my cutoff where my average hazard ratio is about 0.7, around there, how many events do I need? Well, basically what you can do is plug this average hazard ratio. The simplest thing to do is just plug it into the Schoenfeld approximation and just treat it like it's a hazard ratio. And that gives you 332 events. And you can see if the true hazard ratio is closer to 1, your power with 332 events will be pretty drastically curtailed. So, so let's say it's 0.8. So say I had designed my trial to finish in 15 months, my average hazard ratio under these assumptions would be 0.8. So I'd end up needing a much bigger number of events to power the trial if I don't collect information into the tail. Hopefully that's clear. Okay, so average hazard ratio is an estimand. There's a fair amount of literature on this, and I look forward to speaking with LJ later this afternoon about interpretation of hazard ratio or average hazard ratio. But basically, it's estimated by Cox regression, and I'll show you a little more of that. Hopefully this average hazard ratio makes a little more clear what a Cox coefficient is when you have non-proportional hazards. And some people would say it's unfortunate that log rank is the only thing that's widely accepted. So it's the corresponding estimand. I'll claim, but I put a question mark next to this, that if you get sufficient follow-up, the average hazard ratio should be at least fairly stable. Now that there's no guarantee of that, but in what the software I have, both asymptotic approximations and simulation are supported. I'll show you an example. So it's easy to approximate, arbitrary enrollment, failure, and dropout patterns. Cautions, I don't think any single estimand is sufficiently to describe survival differences, much less non-proportional hazards differences. And I'll also caution that early interim analysis should, anticipate possible reduced effect. So I'll keep moving here. So just generally, I just have one slide on this. If you're in that situation where I was testing multiple populations, the prevalence of the populations may be unknown or not what is expected. So you have to anticipate that in how you set up your design. The time to event distribution is likely to be different in different populations. And the overall accumulation of events can occur prior to adequate characterization of the tail behavior. And so those are things that we've tried to anticipate in the way we set them up. So things come up like spending based on multiple hypotheses. There's a nice paper by Dean Folman and others doing this for testing multiple hypotheses in the mid-90s. But we've kind of looked at it as a minimum of planned and actual spending so that we don't overspend at interim analysis. And then in terms of timing, we require both calendar time and a sufficient number of events to accrue before we do an analysis. And so that's just to both ensure adequate follow-up, but then also to ensure not overspending at interim analyses before you get the follow-up you want for the final analysis. Okay, so the asymptotic approximation, I'm still, basically doing the stuff Butch Tsiatis did with group sequential for weighted log ranks. The statistical information is just pretty much proportional to event counts, as would be suggested by Dave Schoenfeld's paper in Biometrics in 1981, or Biometrica. Here we're using a natural parameter of the log average hazard ratio, which is like the log hazard ratio that would be in the Cox model. And the correlation is for the group sequential design are still based on event counts, basically. And so basically, if you're used to the what Jennison and Turnbull refer to as the canonical form in their book, there's just a very simple change from that. And it makes a difference in the design, but in terms of performing the actual calculations, that it's a fairly straightforward change to how to figure out the bounds. Okay, so basically this is the difference that occurs in computing the bounds. In that normally you assume your effect size is constant over time. So this TK is the information fraction at the interim analysis, if you're used to that. So basically the proportion of the final planned events. And basically we're saying that's a function of the TK. So you've still got a Brownian motion that you're looking at to approximate the distribution here, but it's a Brownian motion that doesn't have constant drift. So fairly straightforward. And so the non-constant drift goes back to that average hazard ratio plot. So if you plotted that on the log scale, the derivative of that would just be this theta of TK if you put the x-axis on the information fraction scale. Okay, but your correlation is just the same. It's the ratio of the information fractions. So all these calculations are pretty much standard with this one little wrinkle of non-constant drift. So I expect Cyrus could go back, and if he hasn't done this already, I think he probably has done it already for some of the weighted log rank tests. He could do this. So, previously this Cox model beta hat was shown to be approximately a weighted log average hazard ratio. We'll see this. Anyhow, basically this is all just saying that canonical form, while it's more general than a time to event thing, it could be anything where the treatment effect is changing over time. This is just translating it into this time to event model. Okay, so here's looking at the case that we're looking at that I talked about earlier. So I'm saying I'm going to do cutoffs at 12, 20, 28, and 36 months. My asymptotic approximation says the average hazard ratio is 0.84 at 12 months. 10,000 simulations, say it's 0.839. So if you go down this column, you'll just see these are just spot on. So this is computed with this average hazard ratio calculation I showed earlier. This is computing 10,000 Cox regression coefficients, averaging them, and then exponentiating. And so you're seeing they're essentially identical here. The expected events, they better be spot on because they're done with exactly the same assumptions. They're just, one is computed by, numerically, one is computed by simulation, but they're exactly the same underlying thing. I will not talk about statistical information. I look at statistical information under both the null and alternate hypothesis, but I won't say any more than that for now. Now, in terms of the, having those simulations, I can look at the distribution of the Cox coefficients at interim analysis. You can see they kind of flatten out nicely as you, go through the trial. This is all under the alternate hypothesis, again, with the same times. But if you look at, say, 9, randomly generated trials, this my question at the bottom is really for Cyrus later on. Where do you adapt your sample size here? Because you pretty much enroll everybody by analysis 1, but you haven't gotten your full treatment benefit there. So I'm reluctant to do my sample size adaptation based on what I see there. But again, I'm looking forward to Cyrus's guidance on that. Okay, so spending bounds, pretty simple extension again of Jennison and Turnbull. For lower bounds, I'm going to account for the fact that my average hazard ratio is less. So I'm not going to spend as if I have a constant treatment effect. My early spending probability is going to depend on the fact that I've assumed I've got a delayed effect. So I'm going to have a more lenient early futility bound. So that's accounted for here. Okay, so again, here's my assumptions for the trial. And to LJ's point, I don't know these, but I don't know anything. So, I do the best I can at the time of design, and I try to account for a reasonably conservative alternate hypothesis. So my average hazard ratio at 12 months, 0.84, 20 months, 0.74, 28 months, 0.7, 36 months, 0.683. So for these, based on the assumptions, certainly if I have proportional hazards, it'll probably be of interest here because I'll have, over 60% of my final events. And so probably it's a reasonable place to do an efficacy analysis. Here, I'm going to focus more on futility. This is one, I'm just doing a one-sided design, but basically the interim boundary crossing probabilities are much lower than with proportional hazards. So if you don't account for the non-proportional hazards, you'll be over-optimistic for efficacy. So that's my only point on this slide. The details are not in there. So this one has beta spending for non-proportional hazards. So basically and I won't go through the details, but I'm just using spending, but the spending is under this non-proportional hazards assumption. So that ends up making your futility bound, more conservative. I think this is about a 0.1 one-sided in the wrong direction, if I remember my multivariate or normal distribution. So it's basically saying, well, there's a pretty strong trend in the wrong direction. Now, these you could say, well, I'm well past enrollment. And I didn't put my approximation of what hazard ratio this corresponds to But basically you're saying, you need a trend later on. Since we've already enrolled, we often, would not have lower bounds later. We might just have a single interim at the first interim where I do futility testing for the wrong direction. And we would just delay efficacy for the 20-month, 28-month, and 36-month. And then you'd just be doing kind of a safety bound here at the early one. But that's kind of my personal favorite. I've never really particularly made that anybody else's favorite. Okay, good. Well, we'll have a few minutes for discussion if needed. So the asymptotic approximation for non-proportional hazards using the log hazard ratio is pretty accurate. And we've also done this for weighted log rank tests and combination tests. Cyrus has certainly done those as well in, I don't know if he's done it commercially, but maybe he can comment if he's on. So basically we've built some tools to enable this, and I haven't put up the software, but let me know if you're interested, and I'll try to get you a reasonably current version. So, but the importance for design considerations is you want to time early futility analyses, to be not too early, but then I tend to focus on, is there a strong unfavorable trend? If you have an unfavorable trend, that may not be bad if it's weak, because you've got plenty of time for a strong effect to emerge later. So hopefully that average hazard ratio figure, convinced you of that. But then if you use that average hazard ratio figure, you can also suggest, what might be reasonable times to do your later analyses. So spending, I'll just say again that I think information-based spending alternatives can be useful. Spending by information has gotten us in trouble, to avoid late, overly stringent final efficacy bounds if we overspend early. But also it's important to, in the context when you're testing multiple hypotheses, to just simplify spending so you can test all of your hypotheses at one time. So I've actually done a good job of finishing with a few minutes left over. So if there's no questions, you get a break. And if there are questions, I'm happy to try to answer them. Oh, and I've got a long list of references at the end as well. So anyhow, thank you.
Yeah, thank you very much. So we have a couple more questions in Q&A box. So let me read this. So one is from LJ again. For the delayed treatment effect case, maybe we can ask what would be the long-term survival benefit. So we may choose a landmark time point,
Sure.
Say 2 years, and use the survival rate difference as a metric for the treatment
Absolutely.
effect.
Yes.
Or one may use the area under the curve beyond the 2 years to get a more stable estimate.
Well, I don't think area under the curve is going to be probably more stable than the average hazard ratio. But that may be a debate for another day. Certainly that long-term milestone is very meaningful, but remembering some of the crossing survival curves I showed in the intro, it's not a single number in its own right that's kind of sufficient. And generally, LJ, I haven't looked to see say for the particular example, let's say I'd done a 2-year milestone as the estimate for the assumptions I have. I actually don't know the answer as to whether that would be more powerful under the assumptions I had or less? It's actually a good question. But certainly, that milestone is very interpretable, and people are going to look at the Kaplan-Meier curves, anyhow. If you can unmute LJ, I wouldn't mind hearing his comments on that, but I'll talk to him later about it too. Looks like we have a couple other questions.
Okay, so another question is from Kun Chen. If there is a delayed treatment effect, should we delay the interim analysis for both futility and efficacy?
I think that's something you really need to think through carefully. We did a small molecule add-on to Keytruda for melanoma several years ago, and it looked great in phase 2 studies, but it looked like water in phase 3. And I don't think we had initially built in futility bounds. So there's a scenario where maybe we wish we had built them in. On the other hand, once we started doing a lot of Keytruda monotherapy trials versus chemotherapy and saw, repeated success, we were very reluctant to build futility bounds. Now, part of that was because they wanted to allow the time for treatment effects to emerge. And that's, one of the motivations for using the changing effect size over time to plan those futility bounds. So I hope that's somewhat helpful response to that. You can feel free to send me an email, afterwards if you'd like to follow up on that question.
Okay, thank you very much. So another question is from Tsiatis Meta. Your model is based on Brownian motion with changing drift, but under the null hypothesis, there is zero drift. So why are the group sequential boundary different?
The efficacy bounds aren't any different is the short answer to that. The efficacy bounds are exactly the same, but the futility bounds would change. Sorry, thank you. Good clarification. I hope that answers the question, Cyrus.
Okay, so another question we have is from Yemi. Thank you for the talk. Is there enough data, clinical and empirical, to inform the delayed separation time? For example, 4 months as you use here.
Yeah, it would be good to look at that a little bit further. I think, as I said, we've done so many trials of monotherapy versus chemotherapy that I feel pretty comfortable with it. But, you can complain, usually you don't know the timing delay. So it would be useful to go back and look at, say, ipilimumab to see if it had a similar sort of delay. And I guess they have both lung cancer and melanoma data. That's a good question. But then if you've got a new mechanism of action, it becomes really an excellent question. And what do you assume? I found people maybe excessively wanting to assume delayed effect when based on the mechanism of the new treatment arm, there's no reason you should expect delay. So I think there's a lot of judgment involved in that question. In our case, we eventually developed some data. So I think for immunotherapy, that 4-month period in advanced disease may be kind of a safe, rule of thumb. I love rules of thumb for the time being, and at least to allow you to a little bit of conservatism in your design.
Thank you very much. Another question is about I think it's about your package, our package, GS design and GS serve. So the question is, will there be more development for including strata? So that's a question.
For including strata?
Yeah, stratum, strata.
Yeah, GS design does include strata, but it assumes the same hazard ratio in each stratum. I have a GitHub site, so if you can't find it, it's just Keaven. And under that, there are 3 packages supporting this work. They're not completely up to date, but they should be functioning. One is for simulation. One is for which is just called SimTrial. One is for the average hazard ratio calculations. It's called GS Design 2. And in that GS Design 2, eventually my intent is to put the material that graduates from a 3rd package, which is called GSD-MVN, Group Sequential Design Multivariate Normal is where the acronym comes for. But that is what sets up the non-proportional hazards model. So that includes, in the version that's there, it includes this log hazard ratio calculation, maybe not the most recent update. I should try to update it, but feel free to reach out by email about it. Also includes the weighted log rank. And hopefully there's enough vignettes to make it usable. But, I find that if you don't get these methods into use, they gain no popularity. And that's a large reason for me to, want to support open source design. And then I count on people like Tsiatis to industrialize things for those who, insist on commercial software. Anyhow, so hopefully that's a reasonable answer, but again, feel free to follow up by email.
Yeah, thank you very much. So another question is from Rich Gelber.
Oh, hi, Rich.
The question is, can you identify patient who is going to be progressed within 4 to 6 months, and hence not benefiting from the treatment?
That is the $64 billion question. It was interesting. 2019, the Europeans brought in all the companies that had done these PD-1 trials, and they asked that question. They said, if we combine all your data sets, we think you can tell us which patients benefit and which patients don't. And so each company, getting companies to collaborate and put their data together would take forever. So each of us did it separately. But Merck, we had, I think, 14 trials we could include. And generally, the best we could come up with, Rich, was, people who are at high risk of early mortality are the ones there. But in terms of any specific risk factor, other than something like the, PD-1 biomarkers, which still are not, they're good, but they're not great biomarkers. That's about as far as we could take it. And you can be more sensitive if you want to go to something like tumor mutation burden, but then you leave out probably a big population that has lesser but still important benefit. So that's a question that everybody wants the answer to, and so far nobody's found it. It's not like KRAS, say for colorectal cancer as an answer for selecting treatment. But it's a great question. And generally, we just haven't known the answer up front as to what the subgroups are, so we can't design by them. But the software all supports stratified design, and with the newer versions, you can support it with different hazard ratios and different assumptions on effect size over time in different strata.
Okay, thank you much. I think that's the all question we have, and then also it's time. I think it's at 2 o'clock now. So yeah, so let us close the seminar today. So again, thank you very much for the great
sorry,
talk, Dr.
Sorry
Anderson.
not to be there in person.
I very much appreciate it.
Yep.
And then everybody, please give the big applause virtually. So bye, everyone.
Thank you.